The magic of FGF21. FGF21, otherwise known as fibroblast growth factor 21, was discovered as recently as 2005, and since then it has become a promising area of research. This hormone is at the core of the body's starvation response. So far, it has been implicated in the maintenance of body temperature, fertility in females, circadian rhythms, fatty acid oxidation, glucose metabolism, and possibly even an increased lifespan. We will start with FGF21 and the liver. Hepatocyte cells in the liver are believed to be a major source of circulating FGF21, but FGF21 also has downstream targets within the liver. When FGF21 reaches the liver, it increases fatty acid oxidation, which is the process by which fatty acid molecules are broken down into compounds that cells can use as an energy source. Fatty acid oxidation requires a lot of energy, and FGF21 contributes to this process by regulating the amount of mitochondria present within a given liver cell. FGF21 also decreases growth hormone response and leptin sensitivity in the liver. Leptin is a hormone produced by adipose tissue that regulates energy metabolism by inhibiting hunger.
In addition, FGF21 has been shown to increase adiponectin secretion. Adiponectin is a protein secreted from adipose tissue, and it is associated with an increased insulin sensitivity in both mice and humans. Adiponectin is also implicated in glucose metabolism. Adiponectin stimulates the expressions of GLUT1 glucose transporters, resulting in a decrease of circulating blood glucose. This results in lower levels of insulin and thus mediates the increase of insulin sensitivity. Adipose tissue is a key player in the metabolic effects of FGF21. In brown adipose tissue, FGF21 stimulates thermogenesis, the process by which your body maintains the core body temperature. Thermogenesis is a process that expends a lot of energy, so it can result in weight loss. White adipose tissue is known to mediate lipolysis, but the why and how is not well understood. FGF21 is also known to act in the brain, particularly in the paraventricular nucleus of the hypothalamus, nucleus accumbens, and ventral tegmental area, where it serves to decrease sugar cravings and the dopaminergic reward response. It has also been known to work in the suprachiasmatic nucleus of the hypothalamus and the dorsal vagal complex in the brainstem, where it suppresses appetite in the nucleus of solitary tract. FGF21 overexpression in the suprachiasmatic nucleus results in female infertility in mice through the inhibition of ovulation and is also known to disrupt circadian rhythm. Its actions on the hypothalamus also have downstream effects on the HPA axis, mediating many important functions in the liver. As we've already seen, not everything FGF21 does is good. Unfortunately, FGF21 expression can also lead to an increase in bone mass loss. In the pancreas, FGF21 promotes beta cell survival and protects other insulin-producing INS cells from glucolipotoxicity and cytokine-induced apoptosis, which normally kills these cells. As an interesting side note, FGF21 overexpression in mice was discovered to result in an increased average lifespan from 28 to 38 months. The best part, no dieting required. Many of these findings are very recent and require more research before we fully understand their mechanisms. In the future, research in this area will hopefully lead to life-saving medications targeting FGF21.